Hello student, my name is Chavani SS. Today we talk about the recapitulation of Mendelian principle from your chapter number 1 genetics. So this is our lecture first for the chapter number 1, that is a recapitulation of Mendelian principle, in which we study the work which is carried out by the Mendel, because Mendel is known as a father of genetic. He is a pioneer of genetic branch. So what actually he work did in the field of the genetics, we have to study in this lecture. So basically, what is meant by the genetics? So genetics is a branch of biology which deals with the hereditary and variation of organism. But what is meant by the hereditary? Hereditary means the transmission of character from one generation to another generation. So this character transmitted through parents to offspring. And variation means the same species, okay? Same species carry the differences among them, which is called as variation. So in genetics we study this transmission of character, how it occur in the organism, and what the kind of variation occur in the organism, and how this character transmitted through one generation to another generation. So all this study we studied in the genetic branch. So this transmission of character is done by the chromosome. So chromosome carry the hereditary information. This characteristic information carried by the chromosome, which is also known as the gene. Gene is a coded part of DNA which carry the information regarding to the organism. There may be its morphology, its anatomy, physiology, its physical or mental biology. All these information carried in the chromosomes, and these chromosomes are transmitted from parents to offspring, and this is the hereditary characteristics of the organism. So basically this information are located in the form of the nucleotides in the DNA. DNA is deoxyribonucleotide acid. So this nucleotides containing the three parts, that is pentose sugar, phosphate group, and bases, nitrogen bases, where nitrogen bases are four types, that is adenine, cytosine, guanine, and thymine in DNA, and where the RNA containing the uracil instead of thymine. So these nucleotides arrange the DNA structure, and this coded part of DNA is called as gene, and this gene is responsible for the transmission of character from one generation to another generation. So what kind of actually this gene do? Carry the information in our body system. Basically, the containing the information for the production of the proteins. So, how these proteins are formed? Basically, the process of the transcription and translation. DNA, which containing the coded part of the information, which is known as the gene, this gene is transcribed into the mRNA, that is messenger RNA. This messenger RNA containing the information regarding to the formation of the amino acid. Amino acid are the unit of protein. So this RNA is then translated into the proteins, and these proteins are useful for the building of the structure of cells and the cell function. They may be the formation of enzymes. Enzymes are proteinous, and these enzymes are capable for the accelerating the reaction which happen in the cells. So this DNA is responsible for the what kind of actually functions happen in the body of organism, all regarding to the structure of DNA, which carry the information regarding to the all functions of the cells. So these chromosomes and genes occur in a pair, and these pairs are separated during the gametogenesis, and then it transmitted from the one parent to offspring, which is called as the hereditary. So these chromosomes containing the information regarding to the character of organism. So basically these all the concept which put forward by the Gregor John Mendel, which is also known as the father of genetics. Basically Mendel is an Austrian monk which is born in the Czech Republic in 1822. Basically, he is a son of the peasant farmer who studied the theology and oriented preceded order to Augustine. So basically, he studied the theology and also he studied the botany also. So he went to the University of Vienna where he studied the botany and learned the scientific methods. So basically, he studied the botany and his work on pea plant is a pioneer of the genetic field which he do work about for the 8 years. So he work on a pure line of P for 8 years. So prior of the medel heredity was regarded as the blending process and the offspring were essentially dilution of the different parental characteristics. So Mendel gives the concept regarding to the hereditary. Hereditary means transmission of character, how these occur. So he gave the concept of blending process. Blending process means the offspring containing the two types of genes, that is one from mother and one from the parent. So these different parental characteristics occur in the offspring, which is also known as the hereditary characteristics, and this concept put forward by the Mendel. So that Mendel worked on the pea plants and about 7 traits he studied for the characteristics of the pea plants. They may be their shape, their color, their flower color, their pod shape, the pod color, and the axial terminal of the stem, and also their height. So these 7 traits are studied by the Mendel on the pea plant for the study of the genetic characteristics. So in 1866, he published the experiment in the plant hybridization. So he do work in the hybridization process in which he established his three principle of inheritance. This principle we studied in later on. So basically he tried to repeat his work in another plant but didn't work because of the plant reproduce asexually. So his work he the work he carried on the pea plants are ignored for about 34 years until the 19th century. So basically the three independent botanists rediscovered the Mendel's work, and the Mendel was the first biologist who used the mathematics to explain the result in quantitatively. So Mendel's predicted the concept of gene, that gene occurs in a pair. So basically Mendel is the first scientist who gave the concept of gene, but he didn't know that actually the genes are the part who consider the information regarding to the characteristics of the organism. But he gives the name of this substance who carried these characters from one generation to another generation as a factor. This fact, he said that he predicted that the factors are responsible for the transmission of character from one generation to another generation, and also he put forward the concept that is this factor is occur in a pairs. The gene concept are discovered later on the after the Mendel's work, but he gives the concept of the gene and also he gives the concept that gene occur in a pairs, so that one gene of each pair is present in the gametes. So during gamete formation, the one pair of gene sorry one gene of pair is present during the in a gametes. So one gamete come from the mother and one from the father to form the gamete of organism. So the offspring containing the two types of genes from mother and father, so that he is having the blending process of the characteristics of his mother and father. So basically some genetic terms which put forward by the Mendel's for his work, and this genetic term you should be know. Okay, so basically first of all, that is gene. What is mean by the gene? So gene is a unit of hereditary. Hereditary means the transmission of character from one generation to the another generation. Gene is a coded part of DNA. So it is a section of DNA sequencing encoding a single protein. So gene is equal to one protein. So Gene is a unit of hereditary. It is responsible for the transmission of character from one generation to the another generation, and it is a coded sequence of DNA which encodes the proteins. Secondly, genome. The entire set of genes in an organism which is called as the genome. Whatever the genes we present in the organism's body are called as the genome. Next, that is allele. What is meant by the allele? So two genes that occupy the same position on homologous chromosome that cover the same trait. So alleles means the two genes. So this is the part of the one gene. So gene is found in pairs. So this occupy the same position on homologous chromosome and cover the same trait of organism. And next, that is locus. What is meant by the locus? So locus is a fixed location on strand of DNA where a gene or on its allele is located. So locus is a location of gene on the strand of DNA, which is called as locus. Next term, that is homozygous. What is meant by the homozygous? Homozygous means having the identical gene, one from each parent for a particular characteristics. They may be its height, its color. So these identical genes are known to be the homozygous. They may be the capital TT or so these identical genes from each parent, that is called as homozygous. Homo means similar. Okay, similar or identical genes transmitted from each parent for a particular characteristic. Second one, that is heterozygous. Heterozygous means hetero means different. So having two different genes for a particular characteristics from each parents are called as heterozygous. For example, for height, one is capital T and one is a small t. So that they are two different genes from each parent and for one particular characteristics of height. Next one, that is dominant. What is meant by the dominant? So the allele of gene that masks or suppress the expression of the alternate allele and the trait appears in the heterozygous condition. So dominant means the allele. Okay. So gene occur in a pairs which carry the two alleles. For example, one gene for height. So the tall height, the gene consisting two gene, that is capital T for height. So this dominant gene show the tall height in the plants. So also it occur in the heterozygous condition, that is capital T small t. So this capital T hide or suppress or mask the expression of this small t, that is trait for the small height. So this dominant allele hide the expression of this small t, that is trait of small height. So this trait appear in heterozygous condition. Then what is meant by the recessive? Recessive means an allele that masked by the dominant allele and does not appear in heterozygous condition, only in homozygous. So basically recessive means the allele. Okay, so basically genes are present in pairs and these pairs one gene means allele. So if they occur in a heterozygous condition, that is one is a capital T and one is a small t, then the dominant allele hide the expression of this small t. So an allele that is masked by the dominant allele which is does not appear in heterozygous condition. So whenever they present in a homozygous condition, that is small t, then they appear in their trait, that is small t. But if they present in a heterozygous condition, that is capital T and small t, their expression doesn't occur in that generation because their expression hide by the dominant allele which appear in the heterozygous condition. Next concept, that is genotype. What is meant by the genotype? So genetic makeup of an organism is called as genotype. So this And phenotypes means the physical appearance of organism. They may be its morphology or its physiology. So this physical appearance of organism which we can see. So this may be the genotypic or may be the environmental. So this physical appearance of organism are called as phenotypes. So in today's lecture we see the monohybrid cross. What is meant by the monohybrid cross? So monohybrid cross is a genetic cross involving single pair of gene, that is they containing this single trait. They may be the height, they may be color, they may be pod shape. That is one trait. They consisting just one trait from and containing the single trait from the different parent. So the genetic cross which involving the single pair of gene and parents differ by the single trait. So next one, that is P. P means parental generation. F1 means first generation, that is offspring from the genetic cross, and F2 means second generation of genetic cross. So in this Mendelian work, he do the hybridization method, that is they cross the pure breed variety and form the hybrid variety. Basically, Mendel studied the 7 characteristics of pea for their hybridization process and the genetic concept and his study. So the traits are the stem length, pod shape, seed color, flower position, flower color, and pod color. So this first of all the trait, that is stem length. He containing the 2 characteristics of this trait. Stem length, first of all, he gives first he all these characteristics of this parent are pure line parents. So he formed the hybridization process for this pure line parental trait. So first trait, that is stem length, and he use the characteristics, that is one is tall and one is dwarf plant. Second, that is pod shape, so one is inflated and one is a constricted. Third one, that is seed shape, so characteristic, that is smooth, and second one is a wrinkle. Seed color, that is they may be yellow or green. Flower position, they may be lateral or terminal. Flower color, they may be purple or white. Pod color, that is green and yellow. So these line characteristics are all dominant character of the pea plants, and these all the characteristics belong to the recessive characteristics of pea plants. So all these 7 characteristics of pea plants studied by the Mendel for his study of the hybridization concept. So let's see the how he do the hybridization process for his study of genetics. So first of all, we see the monohybrid cross for the stem length. So basically he used the height of plant for study of the monohybrid cross. Mono means single. Single trait are used for the cross, and he used the pure line of parental plant. So basically he used the pure line of parent. So these are the parental true breeding homozygous plant. So one plant having the characteristics of tall, that is containing the gene, that is T. So one T means allele and one T means allele. So one gene containing the pair of allele, and he cross with the dwarf plant which having the genetic makeup of small t. So he hybridized these two plants and formed the F1 generation is heterozygous, but he having the result, that is all tall plants are formed in the F1 generation, that is T T T T. So all these hybridization occur and result is the all plants are tall. So in this, first of all, the p parents are all he gi- study of his hereditary characteristics of the genes. He used the stem length for his monohybrid cross, that is single trait cross. He used the pure line of parental characteristics. So one is a tall plant and one is a dwarf plant of pea plant. So the tall plant containing the genotype that is T, whereas dwarf plant containing the genotype that is small t and small t. He crossed both these plants and formed the hybrid variety, that is F1 generation, is heterozygous, but the result to occur, that is all plants are tall. But first question is, we use the two line of parents, that is one is tall and one is dwarf, but F1 generation containing the all tall plant. So how this happen? So Mendel used the mathematics for the expression of his work. So he used the Punnett square for the genetic crosses. So it is a useful tool to do genetic crosses. For monohybrid cross, you need a square divided by 4. So it looked like a window pane, and we use the Punnett square to predict the genotype and phenotype of offspring. So we use, for the monohybrid cross, we use the 4 square of Punnett square. So how to use the Punnett square? So step 1 is, that is determine the genotype of parent offspring. So first of all, we have to know the of parental organisms genotypes. So parental organism genotype is capital T and small t. So write down your cross and draw a p square. So cross is, that is capital T and small t. So we have to write here, that is capital T and small t. So like this, we have to write the genotype of organism. So Punnett square, that is fourth step, that is split the letters of genotype for each parent and put them outside the p square. So parental genotype we put on the square of Punnett square. So here we put the capital T, small t and small t. So it is easy to determine the possible genotype of the offspring by filling in the p square. So we can easily find the possibility of genotype of offspring by filling this p square. So how it occur? So first of all, that is capital T, small t. Secondly, capital T, small t. Next, capital T, small t. Also here also capital T, small t. So summarizing this result, we can find that the all genotype is 100% capital T and small t. So basically here we having the one character of the dominant gene and one character of recessive gene. Capital T show the tall height, where small t show the dwarf height. But phenotype of all plants is 100% tall plant, because capital T hide the expression of the small t, which is recessive allele of dwarf plant. So the summarizing result, that is genotype and phenotype the offspring. So genotype is capital T and phenotype of all plants, that is tall plants, because each genotype containing the one capital T, which represent the dominant characteristics of the plant, that is height, tall height. So all the plants of F1 generation are tall plant which having the genotype, that is capital T and small t. So let's do the monohybrid cross for F2 generation. So if you let the F1 generation self-fertilize, the next monohybrid cross would be. So we have to self-fertilize the F1 generation. That is, F1 generation having the genotype that is capital T, small t, capital T, small t. We use the self-fertilization method for the F1 generation for the formation of the second generation, that is F2 generation. These both plant having the showing the phenotype that is tall plant, tall plant, but its genotype is capital T, small t, capital T, small t. So we use the Punnett square for the recognition of characteristics and genotype of the next generation. So here we put the capital T, small t, capital T, small t. The result would be capital T, okay, small, sorry, capital T, capital T. Here capital T, small t, capital T, small t, and small t, small t. So genotype is, that is one plant having the dominant allele, that is capital T, capital T, which showing the genotype that is tall plant, where two plants showing the genotype that is capital T, small type, small t, that is which represent the tall height, that is two plants with tall height but having genotype which heterogeneous, that is capital T, small t, and one if having the genotype that is small t, which is in homozygous condition, which show the dwarf height of plant. So genotypic ratio should be the 1 to 2 to 1. So one plant is a tall which containing the homozygous dominant allele, that is capital T, where two plants showing the genotype that is capital T, small t, that is one is dominant allele, one is recessive allele. So dominant allele hide the expression of recessive gene, and one is the dwarf plant which showing the genotype that is homozygous recessive allele, that is small t. So the phenotype would be, that is physical appearance of the plants should be the three tall plant and one is a dwarf plant. So phenotypic ratio should be 3 to 1 occur in the F3 generation after the self fertilization of the F1 generation. So F2 generation is our 3 is a tall plant and 1 is a dwarf plant in phenotypic cross, and genotypically one is a tall with homozygous allele, dominant allele. Two is the heterozygous condition, that is one is a dominant allele, second one is a recessive allele, whereas one is a recessive allele in homozygous condition. So genotypic ratio should be 1 is to 2 is to 1, whereas phenotypic ratio is 3 is to 1, where 3 are the tall plant and 1 is a dwarf plant. So the secret of this Punnett square, that is key to Punnett square, which determine the gamete of each parent. So we have to use the this Punnett square useful for the determination of gamete of each parent. So how the splitting of the genotype of parents are occur. So without using the Punnett square, we can calculate or determine the offspring genotype. So how? So we have to form the gametes of this parental generation. So capital T, small t, small t. Then the cross occur in those two parents by the hybridization process. So once you have the gametes, so these gametes are put forward on the Punnett square. So capital T, small t, and then we can use the by Punnett square and form the genotype of offspring. So if you have the another cross, that is heterozygous cross, that is F1 generation self-fertilization cross, then we can use the heterozygous with the homozygous cross, that is one parent which having the tall phenotype but having the genotype that is capital T, small t, and one parent with homozygous recessive characteristics, that is dwarf plant. We can use this shortcut for the determination of the genotype, that is this parent containing the capital T, small t, and this parent containing both are the small t. So this capital T, small t, capital T, small t. That is genotype, that is 50% are the tall plant where the 50% are short plant. So there may be genotype is 50% is one is a dominant allele and one is a recessive allele, where 50% genotype is capital small t, that is both alleles are the recessive in nature. So the phenotype of these plants, that is 50% of tall plants which containing the genotype, that is one dominant allele and one is recessive allele, where 50% are the dwarf plant which containing the genotype, that is small t, that is both are the recessive in nature. So let's see another example of this monohybrid cross for the flower color. For example, flower color. So parental, which one is a purple which having the dominant characteristics of this flower, and one is a white which is recessive in nature. So if we cross this homozygous purple, that is homozygous means having the two similar identical genes or allele. So both are the capital P for the purple color. They are dominant in nature with the homozygous white. Homozygous means similar. So for white is a small p. So they are recessive in nature. So for F1 generation, when we cross this pure line parental plants, flowers, and produce the F1 generation. So F1 generation gives the all purple plant with the heterozygous genotype, that is one is capital P, one is small p, that is one is dominant allele and one is recessive allele, where dominant allele hide the expression of recessive allele. So how the cross of F1 generation occur? So we cross the F1 generation by the self-fertilization, which containing the heterogeneous alleles. So one is a capital P, one is a small p, one is a capital P, one is a small p. But both plants showing the phenotype, that is they are having the purple color. So by the fertilization of F1 generation, we split the gametes of this F1 generation. So capital P, capital P, small p. So we form, by using the Punnett square, we can form the genotype of the offspring. So capital P, capital P, capital P, small p, capital P, and small p, small p. So the genotype we get, that is F2 generation having the one with homozygous dominant allele, that is one is a which containing the homozygous dominant allele with for purple color, 2 containing the heterozygous alleles for the purple color, and 1 with the recessive allele for the white color. So phenotype of this generation, F2 generation, is 3 purple color and 1 is a white color. So this is the monohybrid cross of the Mendel's work. So he worked on pea plants and their different traits. So monohybrid cross means we use the single trait for the cross hybridization process by using the pure line of parental generation for the formation of hybrid variety, that is F1 generation and F2 generation, where in F1 generation we got we get the all plants are tall or all plants are purple in color. So which showing the dominant characteristics of plant. But in F2 generation the crosses are different, that is 3 are tall in nature and 1 is a dwarf in nature. So these characteristics are transmitted through the genes, and this is the concept put forward by the Mendel, so that he is known as the father of genetics, because he is a pioneer of genetic concept regarding to the hereditary and variation. So how these variation occur in the plants or organism, and how these characters are transmitted from one generation to another generation, he studied well on the pea plants experiment, and he gives the three principles for the study of hereditary and the variation of organism. So in next lecture we study the principles of Mendelian. So that is for today. Thank you.